beluga. First one of the dive.
Stalk crinoid it looks like, maybe?
Nope, that's a sea pen. That's umbellula.
Really?
Yep.
I feel like sometimes you guys are just making this up. I guess technically everything's made up.
It's true.
Yeah, my word of the day is botryoidal.
Botryoidal?
Botryoidal. Yeah, I need to write it out, see it spelled out.
It's a fun word, isn't it?
Grape Lake.
Grape Lake.
Grape-like, yeah. I think it's Greek.
More Iridigorgia going by.
I feel like half of getting a geology degree was just learning geology speak, like botryoidal. Like the first entire year was just spent figuring out how to even speak like a geologist.
I'll say one of the most useful classes, actually, in retrospect, that I took in college was a course called Bioscientific Vocabulary that just spent a whole quarter going through the Greek and Latin roots of all different types of scientific terms and how to pronounce them.
That's awesome. That would have been so useful to my own science education.
Just went over another little Bathypathes like black coral, in addition to these little bamboos we're seeing. And there's been a lot more sponge debris on the bottom through here.
It's a large one. Large and old.
What's that coral called again? The one that just passed on the left?
It was a sponge.
Yeah, that was a sponge, dead. Probably some kind of dead faria, but it can be hard to tell once they've lost their kind of structure.
A random little stop sign there.
Nice dodge.
Is anyone else getting kind of a high-pitched feedback in their microphone?
Yes. That's a known issue, I would say. So Dave's working on it.
Excellent. Just making sure it wasn't me.
Is it still there?
No, mine just stopped.
Mine as well.
It's from, there's like some
Yeah, I think,
electrical feedback with the winch.
yeah, so that's from me. My apologies.
But we need you to winch. We do need the winch though, so.
I'll just like start SOSing.
Now I'm going to listen for a pattern in it.
Can we get a quick zoom on this little coral right by the laser?
Yeah, sure. Go ahead and do a really quick zoom there, please.
Snap zoom.
Do one little bump up here.
I think this is an Alternatipathes black coral.
You guys got what you need?
Yep.
Pull wide, please.
Full wide.
We're definitely seeing more of them up here, so seem to be getting a little better habit.
Yeah, we might want to start slowing down now that we're at the crest, because we're starting to see a little more diversity.
Sure thing.
Understood. I might just stop and let us swing for a second.
Yeah, sounds good.
Bridge nav, hold position.
There's a couple little bright red guys. I don't know what they are towards the bottom of the screen.
Those are mushroom corals, Anthomastus.
We have a brief second to, if we want to take a look.
Yeah.
Yeah.
There we go. Go ahead and push on in there, please. You want to turn on that porch light there again?
Yeah.
Thanks.
That's really nice. Thank you.
Yeah.
Oh, it's moving. It waved at me. Me specifically, I could tell.
Full height, please.
Full wide.
And more different types of sponge skeletons kind of mixed in amongst the boulders.
Mm. I'll start slowing my pace a bit. Is there anything in particular you guys are looking for at this waypoint, or just want to survey in a bit of a slower pace?
Just surveying around.
Okay.
Of course, now that I say that, everything disappears.
So one. Do you guys want to keep moving or look around here somewhere?
We can keep moving. The coral seems to have all of a sudden thinned out.
Reach
Garage.
nav, 40 meters, 090.
I say that, and then there's one right in the middle of the screen.
It's all good. Go ahead and push on in there a bit, please. Nice little black coral.
This one's very feathered. It's just like a feather.
Yeah.
I think this is Alternatipathes, but it's hard to tell while it's kind of moving in the breeze, so hopefully we'll be able to tell from some of the still images.
Go back and get another shot here.
I've got more zoom.
Yeah, I need to get us a little bit more stable to get a better zoom on it though. Huh. Yeah, it's not really being terribly cooperative.
Blowing around in the thrusters.
Yeah, let's see if maybe up ahead we can find some ones that are a bit more stable. You want to come full wide, please?
Wide.
Thank you.
Thank you.
Yep. And I need to get out in front of you here a bit too.
Big old sponge.
Whoa.
Oh yeah, that's a nice one.
Gonna do a partial zoom, please.
Partial zoom.
Do one without the lasers too. Okay, full wide, please.
Full wide.
Very long bamboo.
Yeah. It's curious how, yeah, some bamboos decide to grow really long and some decide to not. I don't know if it's a conscious decision. It's a different question.
Possibly different ages, possibly different species.
Whoa, 3.
Yeah, I keep seeing these tall stalk kind of dead sponges. We haven't seen any real live ones. So, well, maybe a little bit on the one on the left.
What happened here?
This one looks like it's growing off itself.
They can do that. Yeah, this one has a little live tissue.
Go do partial zoom there, please.
Partial zoom.
Yeah. Pull wide, please.
Full wide.
Can we look to the right? I'm seeing something kind of in the, oh, it's another one of the vase sponges.
Oh, in the distance there?
Yes.
All right, Tim, go ahead and push on in there, please.
Thank you.
Yeah. Get us centered back up a bit. Actually, gonna come partial wide.
Partial wide.
Thank you. And go ahead and push on in there a bit, please. I'll get you squared up on one of the patterns. Go ahead and push in tight there.
Full zoom. There we go.
Hanging in there.
Good. Full wide, please.
Full wide.
I'm assuming you said come down.
All right. So we're basically here at the waypoint. You ready to go to the next one?
Fine by me. Can we go ahead and trigger a Niskin bottle up here before we move on?
Yeah, sure thing. Is there a certain altitude that you like for that?
If we can get kind of low to the seafloor. There's been a couple other species of bamboo that have just shown up, so see if we can pick anything up in this area.
Okay, sure thing. So about 2 meters off, so not to stir up the sediment.
That would be perfect. Thank you.
Yep.
Chris, can you zoom out on the high pack a little bit for me, please? Thank you. I wonder if it's worth just hopping over to waypoint 9 kind of quickly instead of surveying, so we can get up on the shallower, steeper parts by 10 and 11.
Yeah, we can. If Jess is okay with that, we can do that.
Yeah, certainly. We're going to go down slope anyway, so
Yeah, that works for me.
as long as you guys don't want a lot of good visual contact, then
Nah, I think we just shoot straight across.
sure thing.
Okay, straight across to 9, not 10. Yes.
Yeah, I think that's safer.
Number
In
4.
data, sorry, one sec, guys. Which one am I going for? It looks like number 4, if I can see that correctly. Or am I going for 3?
3's already popped, it looks like. I see at least 3.
Yeah, 1, 2, and 3 have been used. Next up would be 4.
Okay, gotcha. Popped.
That was a good one.
Yeah, 3.2 meters off the deck.
And what sample number is this?
018.
Okay.
Thank you.
Yeah, sure thing. Did they use the scoop out of any chance with the mesh on it?
We're looking at the log to find out.
Okay, yeah, sounds good. So sorry, Chris, what's the the heading that we're gonna go?
040.
040, Rog. And then
Ready to start rolling?
Yeah,
Half knot?
half knot. Let's get wild.
Oh, wow.
We're getting wild with it. 040.
Bridge nav, 300 meters, 040, 0.5 knots. That's correct.
I think he said 030.
Wait, did he?
Yes.
Yeah.
Wait, let's see what he got. Oh, yeah, okay. Bridge correction, that should be bearing 040. We probably would have survived, but
Yeah.
Better safe than sorry.
Look at this one.
Let me go.
I don't see any indication of having used a mesh for sample collection.
Uh-oh.
Okay. It's good to know.
It was there, right? Was it there? This. I made that one too. How many things have been scooped? Because someone could have just put it in without noting that it was a mesh.
It's possible.
I'll check, stand by.
It's all out of sorts right now. Look at that.
Where's the other one? Oh. The one that was on the magnet.
It's in the armpit right now.
Otherwise, we're going on a hunt. We got 15 minutes. Let's set off to the right.
Yeah, another sponge.
It's like a bagel sponge.
There's a couple more of these big vase sponges up here.
Can I come down a wee bit there, please?
Just an update on the scoops. It looks like we have scooped 2 samples, one of which being the nodules we collected as a team yesterday, and then the other being a scoop of small nodules collected today.
Oh, okay.
Or I guess it was all today, but you know what I mean.
Yeah,
We got you.
we got you. We're there. So it is very likely that it just wasn't noted.
Yeah.
And we know we have the one you did went into lambda. No,
Yeah,
right?
Lambda. Yeah.
Where did the other scoop go? Like the other sample from scoops?
I'm not sure. It's probably in the starboard pile.
That's what I'm thinking. It has to be. It's gotta be.
The other scoop sample went in starboard bio box E.
Yeah.
So
So
yeah, it's gotta be in there. I gotta check. I'm gonna open it up. I'll make a note. Let me know. Wake me up. There's a chance that Dan won't want to be relieved for dinner.
What is that off to the right? That's a sponge, right? Or a dead
Yep,
sponge?
that's another one of these giant, kind of mostly dead sponges we've been seeing all throughout this area.
I'm trying to not be sad.
I don't think we have any of that. I don't think we have any of that blue mesh. That was the good stuff. We did that one right before we did the vacuum one. The vacuum expedition. Do we still need to get sediment cores whenever we can? Do sediment cores still need to be taken or desired? Doesn't look like it's going to happen.
No, it's probably not going to happen. They'll be
Yeah.
needed if you get the sea cucumber right next to one.
Right, right. You're right.
It's a nice volcanic capstone on top of this knoll.
With a nice bamboo coral growing on top of it.
Biology and geology working in harmony.
Two hearts beat as one.
It's quite a cliff face there. Sort of drops right off.
I understand.
That's something off in the distance on the right there too.
Yeah, from a distance, some of those sponges just look like cacti. Reminds me of home.
I'm glad you said that because I didn't want to. But I was definitely thinking the same thing.
Great, now I want to make a Western here. A Herc Western.
I was going to say, did you bring your G.I. Joe figures?
Yeah, really?
All right, we're going to do a pilot change here. Thanks for the watch, guys. Okay, everybody, we're just doing a watch change over here. People get up on us, pail back again in a moment.
Check. There we go.
Got you. How's it going, 4 to 8?
It's going pretty well so far.
Not too shabby, just had some chocolate chip cookies.
They were immaculate.
I second that.
It looks like we're the cleanup crew for this dive.
It's noble work. Someone has to do
yeah,
it.
somebody's got to do it.
All right, Steve, do you want to take us through our game plan?
Taking us through where?
You want to take us through our game plan?
Yeah.
While we're still doing our handover?
Sure. So we are in the last watch of the dive. We have almost completed the entire trek of this dive. There's a few more points to go, but we basically arrived at approximately the summit depth, 1,800 to 2,000 meters or so. So our plan for the next couple of hours is to poke around one more waypoint, look at one more peak, and characterize what kinds of samples we might want to take at our, between here and our last end waypoint. For example, rocks that might represent some of the geology that's found in the summit area, any other corals or sponges that might be of interest to kind of our habitat characterization objectives, and probably take one more eDNA Niskin, or one or two, by the end of the dive. And then that'll put us right at about the time it'll take to come off bottom, and we'll start to recover during the second half of this watch. But I think we'll recover with our boxes laden with all of our geological and biological goodies to help us better understand what we saw on this dive today.
What kind of goodies do we have in the boxes? Might be a kukui question.
What don't we have?
That's a very good question. So we have everything from cucumbers, sponges, rock samples, eDNA Niskin bottles, and we also have a fossilized coral on board as well.
I'm excited about that.
So am I.
So we will be going down slope for the next little stretch as we kind of migrate towards a little bit of a saddle in between two peaks. We'll try and see what kind of substrates are there. We expect to see nodules, as they have been, pretty characteristic of saddles or gullies that we've seen on the climb up the seamount so far. We've sampled plenty of them, so we probably won't go after those types of collections, but probably rush over to the slope of the next little peak, which will probably have some basalt debris material that'll be useful for our geology team.
Awesome.
But it's definitely been the case that, when we have summited this last little local high, we definitely saw higher diversity and density of corals and sponges than we have seen on any part of the dive thus far. So I'm excited to see what's coming over the next hill.
Awesome. We've got our front row settled here.
Good afternoon, everyone. Okay, I'm on the line. On the line somewhere.
Hello, Dan.
Howdy.
All right, front row. Welcome.
How is it?
Off SBM. Okay.
So the plan is kind of exactly where we're going to head down to waypoint 9, kind of downslope laterally to try and maintain contact with the bottom, and then kind of migrate from 9, just do a quick survey around 9, see what's there, nodules or sediment, and then move directly to 10, where we'll probably end up close to finishing the dive after doing some sampling around 10.
I was doing some light research on the ferromanganese nodules between shifts, so
As one does.
Yeah,
Maybe-
what did you learn?
maybe I should wait until we see some. So
I don't know. I think you should lay it on us. What is a ferromanganese nodule?
It is something that it's a material that precipitates out of the water onto rocks, and these nodules are spherical, and they mostly occur in deep abyssal plains, 5,000 to 6,000 meters. But as if you've been watching this dive, you've seen some that are much shallower, and so we're not 100% sure about the constraints on their formation, but they seem to form in shallow topography, and from the research I was doing, the different ways they can form are interesting, because most of the ones we're seeing, and I'd assume all of them, are hydrogenic, meaning they form out of the water column. But there's been research done looking at diagenetic ones as well as hydrothermal nodules. And diagenetic means it precipitates out of groundwater. And that's more common in those abyssal plains, which we won't see here on Nautilus, but
it's still interesting to see the hydrogenic ones as well.
I thought it was really interesting in Kevin's talk that inside the sediment pockets that you may be getting the diagenic rising from through the sediment there. I was saying a little earlier, I've always felt like those pockets, which we're talking about between the big lobes of lava flows, when we see these round pebbles in there, that I kind of had always assumed somehow they'd gotten in there and the sediment had filled in around them rather than the opposite, growing kind of from within, from the water flow through the sediment itself.
Right, yeah. It's hard to think about groundwater, you know, because we don't see it. But where these seamounts aren't, you can have the groundwater be pretty voluminous, and we can get nodules out of that. And these ones are, unlike what we've seen, these ones are quite vast compared to the ones we've seen on Nautilus.
All right, who knows that fish?
Anybody? Is that a chimera?
It's a rattail.
Yeah, it's a rattail.
Oh, I couldn't hear you, Kugui.
Oh, I had no idea what
Oh,
it
okay.
was.
Oh, maybe I'm hearing Lynette off. Oh,
Yeah.
in the room, sorry.
That one looks like a Prinoids type of rattail. They're always larger than you would imagine. You know, they look small on the screen, but sometimes in trawl for surveys you can get rattails like these, and they actually get quite large.
How big are they normally?
This is an average sized one, I would suppose. Yeah.
Can we paint it with lasers?
So yeah, lasers, cute lasers.
Cute.
Maybe about 20, 30 centimeters in the body. Total length, maybe 40 centimeters.
If you're an adult watching along, 10 centimeters is about the width of your palm, probably. So you can put your hands side by side. Pretty solid fish. What do these eat?
So the grenadiers, or rattails, sometimes they're called, are macroinvertivores. So they are eating small invertebrates in the sediment or on the surface, usually digging around. Very rarely do we see them feed, though. Most of the time they're just kind of station keeping, trying to avoid ROVs, things like that.
I've never seen one eat.
Yeah, once in a while you'll see them gulp, but they usually will dig through the sediment if they find some sort of animal or infauna.
Okay, I gotta go. Sorry,
Yep.
I am.
No, that's great.
I lost the pot there. That's Gibson is so good at his job. He keeps Herc in view even when it's like in the South 40.
Great looking ripple marks again in the sediment.
We're gonna turn and burn here.
Roger.
You know I can't jump off a cliff without turning around and looking at it, so.
Yeah, there's quite a bit here, kind of just off the basement of the
You want
little
to stop
valley.
her up there? That'll give us a few more minutes to look here at the cliff. You want to get clever, you could move her back down the line, I don't know.
There's a
40
coral
meters.
there.
Yeah, got some, that's a
Make her work.
Prinoid, probably in the genus Narella. Got some unbranched bamboo corals here too in the bottom. We got a nematocarcinus shrimp also. We saw a bunch of those earlier in our watch this morning. The fauna's changed a little bit. Right around when we hit 2,400 meters or so depth. This morning we were seeing very different species, and we've still continued to see new species at this site from about maybe 2,100 meters and shallower. There's other families that I expected to see by now that we haven't seen, so that's noteworthy also. We haven't seen tremendously large corals, although that knoll probably had the most largest animals we've seen so far, which is kind of expected because the currents are probably favorable up there for growing large, and the substrates are stable. They're not going to fall over on themselves, corals or sponge, if they grow too big. It's pretty much concrete up there.
If you want to see any close-ups there, Steve, just shout out.
No,
just
I...
poke around here on the ledge.
Yeah, poke around.
Follow the ledge around here for a while. See some critters down here.
And this coral around here, are we just going to assume this is bamboo coral, we assume?
Yeah, we can take a look. Most of them are bamboos. So these kind of unbranched whips here. Although it would be good to check and zoom in on one of these unbranched things, either on the lower right corner.
Gibson, down 10 meters.
Coming down.
There are also a number of unbranched Prinoids as well. And sometimes from a distance they look very similar.
She can come a little faster back down the line if you want. Yeah.
So the one in the right hand side is very suggestive of a Prinoid and has some characteristics already, like
take
this
your
one
auto
here.
head off there. We'll go tail to tail.
Auto head off.
And that thing that's just sticking out right there, is that a sponge? Is that a piece of coral?
Right in front of us?
You
the,
missed
the
it.
one that
It
was down to our left.
Oh yeah, that was a dead sponge. Yep.
Yes.
Yep. We've got some dead sponge material as well from our collections earlier in the dive, I think.
So it seems that there's a lot more dead sponge in living coral. Is there a reason for
Delta
that?
down to 8.
Yeah,
Well,
right.
you know, there's a lot of, there are processes that occur in the environment. You know, dead sponges and dead coral take a long time to break down in the deep ocean, and you know, just the same, you have a lot of settlement of new corals that we're seeing, like many different size classes. You know, many different sizes of corals. Can we look at this one in the lower center?
Yeah, right.
Get a chance.
So I can pull Atalanta and then land.
Are we stopped also?
We're actually bringing the ship back a little bit because I got out of the box there. I can pull that little ROV all wherever we want. It's going to be a bit of a... Look at it with bubble camera.
You can do a zoom with bubble cam, probably get some really nice detail.
Sorry, didn't think I'd come down that far.
And what was that organism that was to the left of this coral that we were looking at right now? It just fell off.
Oh, that was a dead sponge material. Yeah, probably a ferret.
We'll have to do an on-the-fly zoom there.
That's all right, yeah.
Go ahead, Dan.
Sure thing.
We're tail to tail with Atalanta, and I'm pulling on it. That's why it's all.
That's all right. Yeah, I don't want to spend too much time here. I think we'll see similar stuff over at 10, and we can hold off until we get to 10. This is a Prinoid as well. You can see there's pretty good current here. The branch tips are vibrating quite a bit, which is probably why we're seeing greater density of sponges and corals. Yeah, that's good enough.
It's a good neighborhood.
So that gets it. That's probably a species of Narella that gives us probably one of two species, or possibly between two species, based on those just quick zooms to help us decide afterwards what it could be.
So Steve, do you want to talk about like what happens after a dive? Are you making kind of like a, a bingo card, a checklist of all of the species or genuses that we saw? What are you?
It's
Come
Sponge.
down another.
it's
5
a
meters.
bit of a
Down 5.
process. You know, there are scientists who will use the video data and annotate, every single minute of it, counting all the corals and sponges that we see, and making those data available to scientists through databases and things like that. There are those like myself who are interested in the collections and the specimens to try and better understand what the species level diversity patterns are in some of the
Auto heads off.
across the landscape that we're seeing. I can make some pretty good calls based on the video since we've done
I think the 6/8's pulling it back around.
a lot of exploration in this area over the past few years. We've amassed a good number of collections from this monument unit as well as other adjacent areas
You might as
that
well
tell us something
might as well
about
line back up,
go ahead and click in auto head there.
identities of
Auto head
species
on.
based on photos
And you
alone
can,
without
look-
having to be
look at Herc again.
invasively sampling
Since you're
the
already
environment
there.
all the time. So in addition, we are going to be taking those whatever samples we
Let me get
obtain
back on the other side there.
this evening and
I think it's
doing
getting
some
pulled around with the tether
documentation,
here.
imaging
What's that?
some of the collections that come up,
Heading set points here, and it's just trying to come all the way around.
Click auto head on.
and making sure that we provide
There we go.
good notes for the museums that go into collections, for the museum specimens that go into the collections, because once they're preserved, they kind of lose their characteristics of life. They lose their
can turn it
color.
off now. I don't know what's going on there.
I'm gonna try and come around to port.
Sometimes they get distorted,
wraps.
their morphology when they get dried or placed in
One
the
wrap
hull.
in the tether.
Yeah,
So-
don't worry about that.
It's a bit of a process, but
Did you try and
preserve
drive it
them so
around?
they can be
No, I
used by
haven't.
the greatest number of scientists for the longest period of time.
Awesome.
How do you preserve them?
Depends on the need.
Our tail to tail.
But generally,
tail
we
to tail.
preserve things in alcohol, or ethanol in this case,
That was weird.
a very high
Yeah.
concentration. And it basically dehydrates them and prevents any decomposition.
Click 0 on all those
But
blue
depending on
bars
the
there.
need,
Maybe
we
one
can
of those
also
is driving
use
it.
other tools that preserve different types of material like DNA or RNA. If we have a lot of material, we can dry the specimens. That's very effective as well for both morphology, so looking at the structure of the coral, as well as the DNA present. DNA is a very stable molecule, and even when dried, it can be sampled and amplified, sometimes for decades at room temperature dry.
Yeah, go further and further away. How
Sometimes
far-
with varying
how
success,
far
but
can Herc drag out of that?
of course try to preserve things in many different types of ways. Oh, that's a huge base. That was a big something.
Up on the left?
So after they're preserved,
that
do,
one right
do you
there.
send them back?
Oh.
Or?
Yeah.
After they're preserved?
Yeah.
So there's a few different pathways.
Try for a
OET
snap
partners
zoom
with
there,
different
Dan.
repositories that will store and catalog and loan out. Yeah, that looks spongy to me.
Does.
Possibly some manganese coating. Base is at least 10, 15 centimeters in diameter. It's very hard to age these critters. There's based on these types of metrics.
Zoom in a bit more if you want.
Sponges aren't
I'm trying to get
necessarily
it.
like trees. You can't count rings and,
That's
and
what I was
get
trying to
any
get a shot of.
idea of the
Totally.
age of it. Oftentimes there have to be other methods
I can't hold it still enough. But
like isotopes, radioisotopes that have to be involved.
I wonder
All right, thanks.
if,
it's making me dizzy. Okay.
speaking of tree rings, you could look at the crust, and if you know the accumulation rate of it, maybe get a rough age.
Yeah, that is something. crust takes a really long time,
Right.
you know, naturally on the rock surface to accumulate. But you know, like a lot of different types of things in nature, microbial processes can speed up, in some cases, crust accumulation, especially on biological material. And so it's not really well constrained, you know, how certain microbes, for example, might, you know, accelerate crust
Okay.
precipitation processes. So, you know, what one process is on the rock surface could be a totally different timeline on some sort of biological material. Those are open questions.
Click in your auto head now.
Auto head on.
Do me a favor.
Important to
full
keep that in
screen
mind.
thing.
Yep.
We have a question from a chat.
Which
Somebody wants to
tension
know
is
why
not-
Atlanta had placed Argus. Sorry if I
We
already
have
missed
it
the answer.
right there. We don't need it there. I like the big screen. We're going to give up on the cliff here. We chased it about, I don't know, 30, 40 meters there to the west.
Yeah, it looks good. Let's go poke around 9, kind of head that way, and then head up slope towards 10.
Roger.
And
where's 9, the top of the hill there? Oh, roger. Okay, I'm going to have to, so remember when we started this whole thing, we were jumping off a cliff?
Yeah.
I'm going to jump off said cliff and run down underneath Argus here across the flat.
Gonna do a Wile E. Coyote.
Want me to come up? Delta's at 4.
No, you're good.
Okay.
I'm going to come down.
Is that a thing people
It's
recognize?
totally. Yeah, I recognize it for sure.
Yelp.
What was that question about?
Oh yeah, that was a question on our repilot. Why did Atalanta replace Argus is the question.
Because I blew up Argus.
I was
Not
like,
once,
Dan, you
but
want to take
twice,
that one?
just to add injury to insult, or insult to injury, however you say that. Yeah, we were having trouble with one of the thrusters on Argus. So I decided, well, let's try using the other thruster to troubleshoot, you know. We'll just unplug that one and plug the other one in. They're electric thrusters. They run at some crazy lethal DC voltage. And we plugged the other one in and it let the smoke out as well. So the controllers are, they're kind of spendy and take a long time to get. So we've been waiting for parts. We finally have the parts to replace the controllers. We just haven't had the time. It's a bit of a involved process, opening up the 1 atmosphere bottle and replacing the controllers in there, which are all, of course, hardwired in, and they're heat sinked to the chassis, so it's not just it's not a real quick fix. And then we have to test it and figure out why did it blow up in the first place. So, which we think we know. It all comes down to one of the cables running to the thruster was probably shorted out. Technically, that should not happen in my defense, but I won't do that again.
When is that maintenance scheduled for?
TBD. It's opening up the 1 atmosphere bottle is not desirable while we're at sea. It's a big, long, 2-meter long thing full of sensitive and expensive electronics, so opening that up at sea is risky for several reasons. But Argus should be back in action for 2023. It's been really good to get Atalanta out of storage and give it a good run. You know, we've made some improvements on Atalanta. Yes, ma'am. Please.
What are some of those recent improvements?
The rotary actuator that operates the camera is it had some issues. It had a built-in compensation assembly that we've replaced with our own custom. We've basically plumbed it to the Atalanta main compensation system. So we custom made some parts, and we've done some improvements with the software, the control of that actuator, so we can so Gibson can actually control the mini Zeus camera that's looking at Herc. So that's gotten a little better. And we've cleaned up some of the wiring on it and replaced some of the components that have just, you know, are getting a little tired from sitting around for years. I've tuned up the lights on it, got the lights pretty dialed.
I have a brief or a small course correction request, not immediate. Can we head to kind of like 100 meters west of waypoint 10 and kind of aim for that spot? So we're kind of more on the western edge of this lobe that's sticking out. Yeah, something like that. Can we go, yeah, to that point? So the logic behind that is that currents have, you know, are predicted to be coming out of the north here. So I figure if we stay out of the shadow, you know, which is on the south side of, you know, kind of these promontories and ridges, we might be able to get kind of a wraparound flow that might be better for things like corals and sponges, but still be able to get the rocks that we need in the slope.
I like that hypothesis.
Doing science.
Doing science.
Now we got to test it.
Right, well, we're on this awesome sand flat that makes me think of the beach. Let's do some introductions. How about your name, your seat, your hometown, whatever you claim that as, and if you had to be a coral, which coral would you be? I don't know. So what's your favorite worm question? I like that one.
Can I be a rock instead?
I mean, maybe. There's no rules to the worm game.
You could be a rock.
Could be a fossil coral.
Yeah, you could be a fossil
It's a
coral rock.
Okay, I'll be a limestone.
Okay, do you want to start?
Sure. So I'm Brandon Scott. I don't know, what would you call this seat? Seat 2 in the back.
Science.
Science seat. And my hometown's Las Vegas, Nevada. Lived there all my whole life. And if I could be a coral, I'd be a fossilized coral known as limestone.
There's this thing down here.
Unknown.
Looks like it's disrupting my nice, peaceful Zen ripples.
You use Zen sand.
You know what these Zen ripples need?
There's something swimming there.
They need they need a Herc footprint.
Oh, yeah.
What is that?
Is that the crazy chicken, headless chicken monster that Liz was talking
Go
about?
for it. Yeah, it's
No,
a sea
it's
cucumber. Wow.
something smaller.
Something. Can we follow that?
Yeah.
What are
Dan, zoom
you?
in just a little bit for me. Not too crazy or I'll lose it. Yeah, a little more maybe.
For everyone just tuning in, hello. We've prepared to find a floating or a swimming little, I don't know what it is.
I know what this is, and
Okay.
I've collected this before.
Yeah, that's good.
And I made a mess of it, unfortunately, because it's very fragile. But this is actually a free-living flatworm.
Cool.
Yeah, it's a type of flatworm, so it's not related to like the polychaete worms and the bristle worms and we usually see. It has these very, very branched kind of digestive tracts. That's what you're seeing, those lines and branches. But it's a flatworm, mouth is on the bottom. I don't know what we're looking at now, whether it's the top or the bottom. But we did sample one of these on a previous cruise last year that I was on. And we didn't know what it was. It was on a rock, so it's good to see that it inhabits this soft and hard substrate environments. But
It looks like a leaf just tumbling.
And if you hold one zoom, it's easier for me, then I can move the ROV closer. when you zoom in, I can't keep it in the picture because it's then a much smaller area on the camera.
Gotcha.
And it moves out like that. So if you keep the lasers in the
I like
picture,
the dendritic internal structure.
then I'll move the ROV closer and you'll get a better picture to focus on.
This gives you a good view of the current, hey? Like, ripping along. If you're a flatworm, it's ripping.
It's up to Steve.
Yeah, looks good. Any more zoom, or are we?
We can, if he'll hold still for a minute. Okay, Dan, get a tight zoom there.
Max out there.
Roger. I can get a little closer if you want to get the full.
Yeah. I don't think we're not going to collect this critter, but
okay, go show me the lasers again, please.
Wow. Dr. Sako Matsumoto says she's never seen one swimming like this before. That was really cool.
So I think we're looking at the kind of the top side of it, or aboral side. Mouth is probably underneath.
What are some other flatworms people might know?
Max out.
Wow.
You know, most of the flatworms are, well, I don't know if it's most, but flatworms are largely known for being parasitic, but there's a number of free-living ones too. They're very small and inconspicuous. They're very simple body plans. They don't have very developed, organ systems. All their tissues are, gas exchange and nutrients exchanged, by diffusion across its tissues. So to see them so large is unusual because it's usually a detriment having a large mass. But they do feed. They have a mouth on the underside and kind of a two-way digestive tract. So food is going in and out of the same hole. But yeah, they're pretty neat. Sometimes they have eye spots. I don't really see conspicuous eye spots for this one.
Oh, there it goes.
Bye-bye.
Yeah.
Bye.
That was interesting.
Yep.
What do you leave behind
It's a
there?
learned experience from the sea cucumbers.
Okay, you can go full white.
All right, great. Let's move on. Highlight achieved. Check.
Check. One highlight.
Hey, look, there's a Herc footprint.
Very nice.
Hercules was here. I'll be there.
So yeah, I
1,000 years.
sampled one of those last year, so we have a bit of it back at my lab in Boston University. I don't know if we sent it out to anyone for identification yet, but now that I've seen more of them, I'm probably more likely to.
Hey, Gibson, come up. Come right up.
Coming up.
Come up a little quicker. Just gonna take this opportunity for some tether.
They're very fragile though. Just transferring it from
Delta?
one bowl of liquid to another bowl just disintegrates. They don't do well when they're taken out of their environment.
Gotcha.
Most organisms rarely do, I think.
Mm-hmm.
Just like me on this boat.
Oh no, hang in
Functioning on 3 hours of sleep. No big deal.
I was like,
We've barely gotten started.
It's dive one. Oh no.
It's okay. I'm good.
Okay.
All right, I'll keep going with the introductions.
Is that a dead sponge?
Yep, it is a dead sponge.
Do a quick zoom there if you want, Dan.
Is one of the creepy spine ones?
Yeah, one of the Prinoids. Yeah.
Are Prinoids the ones that look like rib cages?
Yes, yeah. Yeah, so that's probably the stalk, the vertical part.
Gotcha.
Yep.
Looks like a...
Prinoids are super diverse.
Looks like a spine, some kind of spine.
Yeah. Super diverse family. They can sometimes different species in the genus Pharea can form reefs, actually. We saw that off of Southern California a few years ago, where they were forming large continuous reefs with lots of rubble and debris, and just the living part is just like the very outside top layer. Sometimes hundreds of meters long and 100 meters tall, or like crazy reefs. I've never seen like that before.
Is it common to see that?
Hmm?
Is it common to see those types of reefs?
Not usually, no. It's probably a very productive spot. I think that was south of the Channel Islands, if I recall.
You're pretty tough to hear, at least for me, Kukui. Can anyone else hear well? Is it my mic?
No, you're
Can
quiet.
you scoot your mic like a little closer to your mouth? Give it a try.
And eventually you're going to make your heading 0, whatever the ship's course over ground is.
Okay, how about now?
Better.
Yeah,
Improved.
better.
Thank you.
You're welcome. Sure, Steve. Do you want to keep
035,
doing
it looks
intros?
like.
Yeah.
Give a wave. This camera's on channel 3. If you're watching on Nautilus Live, check out channel 3. If you're watching on YouTube, go over to our channel 3 stream. And you can see Steve waving at you.
Celebrity.
Celebrity, Science Steve.
My name is Steve Oskarich. I'm the watch lead for the 4 to 8. I'm also the biology science lead on this expedition. My job is to make sure we accomplish our science goals for the dive and for the cruise. My position, I am a postdoc at Boston University, where I study deep water coral biodiversity, biogeography, sea mountain ecology, among other things.
Can come down.
Right now, well, my hometown is Milford, Connecticut, southwestern Connecticut, the shores of Long Island Sound. But I try to get back there once in a while, but been moving around a bit the last several years. Let's see, what was the last thing? Your favorite coral, right?
If you had to be a coral.
If you had to be a coral.
It could be your favorite.
I don't know.
So many
Kind of
corals.
creative questions.
Is this like
stump
choosing
me.
among your children?
Yeah.
We could be limestone buddies.
It's trying to convert you to deep geology.
I'm trying to like anthropomorphize like corals and like what their characteristics would be.
You could just
I'm gonna
pick one
blank
you
here.
Choose whatever your heart desires.
I'm gonna go with stylastrids, lace corals. They get no love. You know, they're kind of like
And what's
the
coolest
least
about them?
well thought about coral.
It's okay, Steve. We love you.
Yeah.
All right, so raising the overall public image of stylastrids.
Yeah. So stylastrids PSA, they exist.
Aye.
They just want to be seen.
Come down another 5
Actually, they
meters.
can form pretty amazing reefs in the Southern Ocean, and they're one of the few corals that outcompetes a lot of other coral species in oxygen minimum zones. So they're survivors.
They're really good at what they do.
Mm-hmm.
That's awesome. Beautiful. I think that's an excellent choice.
Yeah.
I think so too.
I would have gone more in depth with limestone if that's what I knew we were doing.
Don't worry, we'll come back. We will tap into all your limestone
You can
knowledge.
get some extra credit on that score next round.
Tilt your camera down a little bit.
Awesome. Kukui, you want to go?
Oh, yeah. Aloha kako, everybody. My name is Kukui Gavigan. My hometown is Kulamoe, but I go to school at the University of Hawaii at Hilo, and I am a data logger. And so I'm logging all the data, everything that we're seeing, everything we're talking about. And if I had to be a coral, I think I would be one of those bubblegum corals. Yeah, they just look super cool.
Good
I
choice.
love that you chose a deep sea coral. I feel like we're converting you on like the first dive.
Yeah, you don't have
I've
to.
been converted.
Amazing.
You can pick shallow water corals.
There's no rules.
I mean, they're pretty too. I like
Yeah.
those.
Bubblegum is a good choice.
Thank you.
Matt?
Hi, I'm Matt. I'm a student intern or like a guest scientist intern. I'm kind of over here in the little corner of shame.
Oh, we can see you.
Oh boy, hi Mom. I'm from California, from San Jose, and if I have to be a coral, I'm gonna go with bamboo coral, just because I like how they look and how they sway in the currents.
Flexibility, I like it.
Yeah.
Good. I'm Megan Cook. I am a Science Communication Fellow on this watch in the afternoon, and I am the Director of Education Outreach for Ocean Exploration Trust full-time. My role is to help facilitate the conversation, share ocean exploration with all of you watching online, so thanks for being here. Send in your questions on nautiluslive.org. My original hometown is Boise, Idaho, Great Potato State, home of so many oceanographers. And I now live in Friday Harbor, Washington, much closer to the ocean. If I had to pick a coral to be, I think today I would be a mushroom coral. I just think they're super cool looking. They, for folks watching online who haven't seen one yet, they are bright red. They don't branch like the others. They look a little bit, when their polyps are pulled in, they look a little bit like a drumstick, or like a almost like a toadstool. They're red and the polyps kind of tuck in and they look a little bit white, so almost like white dots. But when they're out, they have really prominent polyps, and I like that. You know, you can look at them and you like very clearly know what's happening there. You're like, you're a coral. You are snagging things out of the water column. They get pretty big, which I think is pretty cool. It's my choice. Beautiful. I like it. Lynette, you ready for an intro?
Sure. I'm Lynette. I am in the navigator seat. I'm originally from Chautauqua, Wisconsin, very small town. I had a coral picked out, but I looked it up really quickly, and it's actually a sponge. Are we allowed to choose a sponge?
Sure.
I think Walteria.
Okay.
Yeah. I think they're super cool looking. They have this sort of like cage-like structure, and often have like little shrimp friends inside. So it's cool.
Awesome. I love. What first led you astray in thinking it was a coral, or did you just Google it to double check?
I just Googled it to double check.
Yeah. Love it. I appreciate your commitment to accuracy in the introduction question. All right, Dan, you're up.
I'm Dan, sitting in the Herc chair, originally from San Diego, California, currently a resident of Oregon, nowhere near any town. If I had to be a coral, I'd probably be a cup coral hanging out on a cool cliff overlooking a garden, somewhere where a manipulator couldn't get me.
You could live a lot of cool places as a cup coral. Urian Gibson.
Gibson Clark here in the Atalanta pilot seat. I'm an ROV intern, originally from the Neuse Bay, British Columbia, Canada. And if I had to be a coral, gosh, I kind of want to drip the seafloor. I like the looks of that flatworm a lot more, to be honest. So can I change the species and stick with the flatworm?
I love it. One person choose rock, one person choose sponge.
These kids,
is it because you want to travel?
Nowadays.
That's right. See our daisy there, Dan?
Looks like, yeah. Looks like we're headed south there.
All right, Dan, what you got?
Roger.
Alrighty. So I am Dan. I'm all the way here in the corner, and I'm the video engineer on the 4 to 8 watch. And I do things like control all the cameras you see, from on the ships to on the ROVs, straight into your home.
She doesn't.
Living in Los Angeles right now. I'm
She
originally
doesn't
from
let us know
Minnesota.
what's going on. So
And in addition to being a video engineer,
no,
I serve as the
we'll just
expedition
let her-
documentarian.
just keep an eye
aboard
on it.
Nautilus
Yeah, let her
Lines,
do her thing.
documenting the days and lives of everyone on board. If I could be a coral, it would probably be a fire coral, because it's one of the coolest ones I can remember. And Steve, I know you gave us a bunch the other day, but I am really good at not remembering scientific names. So, fire coral.
Still a coral. That's what matters.
One up me, Dan.
I'm in the running.
All right, so what's the dark stuff that we're seeing here, Steve?
So it looks like we moved into kind of these pockets of nodules. They're not necessarily as dense as kind of the nodule fields we were seeing earlier, but as we get closer to this knoll, I suspect there's probably wraparound currents here, which might be why you have more exposed, you know, more favorable nodule terrain. You know, some of the sediment's probably scoured away and you're not seeing as many ripples anymore. What was interesting to note is that through this saddle, the ripple lines in the sediment were oriented northwest, southeast, which suggests the flow is, you know, probably coming off the north or north-south flow around this knoll that we're headed up. So I'm optimistic that that there's going to be some good stuff up here biologically. But as to why the nodules aren't as dense as other sites, I'm not sure. Here, it's probably still a fair bit of sediment accumulation in this channel.
Let's head north for a while.
And where does all the sediment come from?
See if you can go sideways and really work the bow thruster.
So most of the sediment we're seeing here is probably buildup of small planktonic animal shells that and there's not a lot of land sources around here, so this is not sand, this is not necessarily eroded material from, you know, coral reefs or anything like that. Probably small foraminiferan shells that live up in the water column, and when they die, their shells sink to the bottom. The sediment here kind of looks like small white orbs, or yeah, once you remove all the very fine silty stuff, which could be, you know, detritus or other, you know, very fine-grained sediments. It's pretty uniform in shape and size.
We have a question for the chat for the ROV pilots. Do you guys ever listen to music while you drive?
Just Dan's humming. Otherwise, no. We're talking back and forth, making sure I'm aware of Dan's movements and he's aware of mine.
While we're driving what? Driving the ROV?
Driving ROV, I believe. They just said driving.
Driving. Yeah.
Look at this.
So I deviated there for
Look at
the rock.
Flow pattern around that rock. There's probably some current coming in from the top, from the north, and wrapping around this and creating some interesting palm-shaped ripple marks.
That's like a mini primer in sand ripples right there. You can see the bending.
Reminds me of a trilobite,
Yeah.
the shape of it.
There's also an
It's
anemone
an
on
awesome
there.
anemone. Yeah. Big orange
Nice,
anemone.
Anemone or
sand ripples.
maybe Corallimor varian, can't quite tell.
What is the second option?
The second option, Corallimor varians are
anemone relatives, but they
Pull up the DSC
the
camera
sexton
over
up,
there.
they're usually, well, this one's might be sucking
Remind me,
in.
Dan.
They're
Just
usually,
go down.
they have a different kind of internal anatomy, so they're placed in a different group. But this one, usually Corallimorpharians have knobby tentacles, and they typically have a broader oral surface,
Can zoom in there
kind
if
of like
you
this
want,
one.
Dan. It's framed up in the DSC for you too, Steve, if you
Oh,
care.
yeah?
Just a rock. I can turn on the porch light.
Yeah,
while I
so
get
this
it
might
better.
be a Corallimorpharian.
I'm going to turn on another light here.
Tried to collect these in the past, and they kind of, they're really stuck there. Surprisingly, don't want to move very much.
Zoom in
But
a bit more if you want.
this one's been here for quite a while, and I think it's on a pretty good rock. It's not going anywhere.
What a cool shot.
We haven't seen too many anemones on this this dive so far. We saw a few down deeper. I would call this a Corallimorpharian. The knobby tentacles are loaded with nematocysts and stinging cells that the animal will use to capture prey.
We have another question from the chat for the science team. I'm sorry if I butchered this, but are the foraminiferan shells made of calcium carbonate?
Yes, that's right. All right, we're all set back here.
Roger.
Yeah, they are calcium carbonate.
A few more
I'm not sure of
rocks.
the exact mineral, though. I think it might be calcite or calcitic.
Yeah, it's
Which
pretty sure it's
explain,
calcite.
it
Yeah.
would explain their accumulation here, yeah.
Calcite or aragonite are your two options with calcium carbonate.
Somebody say rock? You want to pick up a rock? Is that what you're talking about?
Good rock, bad rock.
Any, we want to go a little bit more for rocks, or do any of these look like candidates? They look kind of rounded to me.
I would go farther if we can. This could be debris flow as well. And we'd want to know where it came from exactly if we can.
Right.
Just like that big rock that the anemone was on. Probably came from upslope. Who knows how long ago?
Hey, you have a lot of options when we're talking in this like 70 to 115 million year range that we're trying to constrain here, hey?
I'm going to shorten up the timeline a little bit there. We got about a half an hour of bottom time left.
All right.
We're probably at, what did we say, off bottom at? Yeah, off bottom at 5:30.
All right, let's try and get to the slope so we can pick up some things. Oh, let me do it. Snap zoom on this pink coral as we fly by here.
Sure.
What type of coral do you think this is?
So this is a precious coral, or hemicorallium. I think it's the first one we've seen.
Right. Keep the lasers in the picture
so
for
far.
me, Dan, when we're doing the flying.
It looks a little ragged. Some of the branches on the bottom are a little broken
general.
off, but this is one of those precious corals. So it's fairly rigid skeleton. It has a snake star, type of brittle star, on as an associate on the colony. They get quite big, and they can sometimes form reefs. Coral gardens, but dense enough that it could be construed as reefs, but definitely coral gardens in some spots, like we were seeing last year up in the Voyager seamounts. Saw some pretty dense gardens of hemicorallium.
See this gelatinous stuff to the left behind the pink coral? Is that just sediment?
Oh, yeah, yeah.
Spider web.
Looks like a spider web, yeah.
Yeah.
I don't think anyone really has a good answer for what that is.
Okay.
It's probably deep sea mucus of some type.
Yeah, when you do those on the fly zooms, if you zoom in till the lasers are just at the top of the screen, does two things for us. One, it keeps the lasers in there so the scientists can get a scale if they're taking stills back there, which they are. And it's a comfortable zoom level for me to be able to move the ROV closer to get a better image. So the closer the camera is to the whatever it is we're looking at, the less seawater we have to look through, and the better image you'll get in the end. So when you do that kind of half laser zoom, I'm still flying the ROV and trying to get as close as I can.
We have another question specifically for our ROV pilots again. How long did it take to descend for this dive? How long do you expect it to ascend?
Like forever. Lynette did
That's a learning question.
our navigator, Lynette has done the math for that.
Yeah, we come up at a rate of about 15 meters per minute, and we'll be ascending from a depth of about 2,000 meters.
Do that laser zoom there if you want, Dan.
So we're expecting it to take about 2 hours and 15 minutes to reach the surface.
We can keep going. We've seen this sponge.
Yeah, I'm just going to
watches.
pirouette around it on the way.
This is Poliopogon. It's a glass sponge.
Turn my joy gain down, I won't be so...
Sampled extensively in this area. I don't know if we have a species name yet. It's a slow process to identify some of these sponges. We have to look at their spicules, which requires microscopes and imaging and a whole process. And then there's only a handful of experts in the world to tell some of these different species apart.
Is it a super species group? Are there a lot of different Poliopogon, or do we not even know that yet?
They have from what Chris Kelly was telling me in previous years, Poliopogon is kind of a catch-all genus.
Okay.
So there's a lot of mishmash of things there that probably don't belong there. But
Okay.
since no one's really specialized in teasing them all apart, it just kind of accumulates lots of different species, or possible different species. So I don't think that's been worked on enough to really tell if it's speciose or not, or if it's just an accumulation of a lot of different animal taxa that scientists have thrown in there over the years.
Gotcha.
But Poliopogon is a genus found worldwide, Atlantic, Pacific, usually quite deep to 2,000 meters or more.
We have another question from the chat, specifically again for the ROV pilots. Why does it take so long to go up and down? Is it a decompression issue?
We have to actually operate Herc through the water column using our vertical thrusters. And maximum speed that can achieve is about 15 meters a minute. Maybe. We can get 25 on the way down. The vertical blades on the thrusters they have more of an aggressive pitch to thrust down as they do up, because they're actually modified boat props. But usually coming up, we're loaded with samples, rocks and such, and yeah, that's kind of our...
What boxes do we have wide open? Or
wide open, we don't have any boxes wide open, but it seems like we have a spot in box B, but it has a sponge in it.
Yeah, maybe we'll double up here.
I think we can also fit something in lambda.
Is that where the sea cucumber is?
No, the sea cucumbers are in the slurp jars over here.
So there's nothing in here?
There's one nodule from Omega that kind of landed into that box.
Okay. All right.
And what glass sponge are we looking at now?
So the one immediately in the foreground is probably Tretopleura, Tretoppleura. It has some critters on there. It looks like an isopod right there. Yeah, and a crinoid.
Mm-hmm.
Starting checks, Dan.
You can let it go.
Roger.
We're
And then
coming up.
there's
You can
there's
do a
some
quick strafe.
Prinoids here too, unbranched Prinoids.
Good with that porch cam.
What's that?
Can I take the porch cam?
That's a
Yeah, you can
tripod
just do a quick
fish.
strafe. We're coming off bottom in half an hour, so.
A tripod fish?
Tripod fish there. Yep.
Oh yeah, cool.
Good eye, Steve. I still don't see it. Now I see it.
Anything looks
Now I see it.
looks angular and
Dan, you want to do the laser
attractive.
zoom for me?
for picking. Stuff here looks pretty rounded to me.
Wow.
Still.
Weird.
It looks like a tiny mololo fish.
Mm.
What's that in its sides that are poking out? They look like feelers almost?
Yeah, they're modified pectoral fins that they're now standing, you know, kind of tripod legs. And then as you get closer, you'll see little rays that are feelers that detect motion in the water, current. And these fish point up current. So we use them like little current meters as well.
Check out his reindeer antenna.
Mm-hmm.
Nice. So there's I think 3 or 4 different genera of these tripod fishes.
Push in
From
just a little bit more if you
I think
want.
they're all from the same family, Hypnoppidae.
Oh, that's a beautiful shot.
That's a super nice shot.
The differences are subtle.
Yeah, it almost blends in with the sand, but you can see those like appendages.
You got him on a DSC too, Steve?
Yep.
I don't know what that'll do.
Yep.
Maybe we can.
So
We
cool.
saw one of these a bit earlier, so I got some images. I'm ready to go when everyone else is, so we can go keep looking for this rock.
Roger.
That's a nice DSC
Looking for
view.
the rock.
Was it on this team's watch earlier when we saw the flip-flop, or was it on a different watch?
It was this watch.
Did we get a picture of it?
We did.
Awesome.
Bamboo coral.
Someone from the chat asking again, what kind of fish is that?
It was a tripod fish. And there is a mushroom coral.
All right.
Yay, it's me.
Oh.
Well, you guys get all the cool stuff right away, huh?
You want a shot of this one,
One
Steve?
of the coral Kardashians.
No, we can keep going.
Roger.
We saw one a bit earlier.
All right, that's my coral of the day.
I want to make sure we get a nice choice rock.
Mm-hmm.
Roger.
Whoa.
Sure.
It's a great view on channel 2. In the Argus view, you can really see the sediment ripples are quite deep enough that they're casting shadows as the Hercules are coming through.
Another question from the chat, specifically for the science team. What kind of pressure are these organisms surviving under?
What are we at now for depth? 20, yeah, so roughly
pressure's half the
200 times
Yeah.
atmospheric pressure.
And viewers can do a little math, calculate it out. 1 atmosphere, 14.7 pounds per square inch. So every postage stamp surface area of you, this accumulated amount of atmosphere at sea level on you is pushing in 14.7 pounds of weight or force. Look at that sea cucumber.
Is that a sea pig?
The sea
It's a
cucumber.
sea
Sure.
cucumber, yeah.
And then every 10 meters of water, so you can have an entire atmosphere of air equals the 10 meters of water pressure adds an additional atmosphere. So anytime you can divide our depth by 10, and that'll give you the total amount of additional atmospheres pressure on you.
It's about 2,911 psi.
Come on, you can't do meters in PSI. You're going to do meters in bar or feet in PSI.
Good call. Good catch.
Oh, there's a formosoma or a
That's just not right.
pancake urchin too. There's a bunch of stuff in this area, but unfortunately, I'm not seeing anything very angular at all. It looks
No, yeah, it's all rounded.
very, very crusty. It's that unbranched Prinoid again.
Come down, Mike.
I'm
Come
coming down.
Tim.
I was just rushing to get out of the.
Go to Dodge.
It was in the circle.
I
Yeah.
don't think it's going to get much better as we get up top either. It's probably going to get even more smooth it out. So, I mean, there's always a chance we can break something off here, but I'm not sure I see anything that might yield that basalt we're looking for. Dan, your mission, if you choose to accept it, is to find a loose rock.
Roger.
Oh, where may we find such a thing?
This is like speed rock selection for you, Brandon, hey? You're like,
I'm trying, yeah. I'm surveying the whole area of the screen.
before someone comes in to relieve you for dinner. Let's find the
If
rock.
they get the rock, we lose. I don't know, that's one of the suspicious ones. What do you think?
Yeah, we can see if it moves.
Just need to go big.
I'm not optimistic, but we gotta try.
Some rock is better than no rock.
See that rock
Oh,
right
That
there?
one's loose.
How
Huh.
about that one?
Oh my.
It's got some corals on it, so
Veto.
Come on, Steve. Let me
now,
get one
I
of these
regret
size
putting
rock.
the camera on the front porch.
Oh no.
Yeah, no, that's too big.
It's quite large.
What you talking, Willis? That'll fit in the box. It's not very big at all.
We don't have any really any big box space left.
What
There
box
may
you got?
be some space in Lambda, but
Oh, it's
there
heavy.
may be one
kinda big.
module in there.
That's a little.
Okay, what's the other little rock you were looking at? This one way up here?
Yeah, that guy.
Oh,
So close.
Oh, that's embarrassing.
We actually have a question for the ROV team. Are you guys free to answer?
Sure.
What is Atlanta's job? Is it more of Herc's supervisor or like a buddy system?
Sorry, I missed that question. Say again?
What is Atlanta's job? Is it something like Herc's supervisor or more of a buddy system type method?
well,
It's a perfect question for Michael.
I ain't knowing. Maybe for Dan, it's a supervisor. Keep Dan on a leash.
Literally.
It's more of a buddy. It's all to support Herc. So to give Herc pilot another perspective on the
Maybe this
location
guy.
of Herc,
Mm,
and to
I
but it
don't see
also
it.
to
Over
takes
here
the
too.
waves. So
Never
if,
know.
Herc would maybe
maybe
bobbing up and down and stuff like that. So it's all more of a buddy system
Also
to
pretty
support
big.
Herc.
Yeah. Or maybe, oh, this guy looks actually pretty nice, if that's loose.
Could be. Where are you going, Herc?
Pop, 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 pop. Although right now I am very much missing Argus. No,
Yeah, me
prefer Argus over Atalanta.
Why is that?
Argus is bigger and could take the up and downs
You got your
a lot
choice here.
better.
1, 2,
Heavier.
or the little guy in the background. Oh, nope, that one's stiff.
I guess the big guy.
First one?
Well, can you pick this one up?
Meanie.
The
Meanie. I could get that one.
yeah. Let's see. Let's look at it.
Kind of flattish. Crusty on the bottom.
Oh.
It look more like a cooked potato. I would say that one's got some more angle.
Yeah, this one looks better. If it can fit.
It can fit.
Oh, it'll fit.
You can put it in the 4 bio box on the left-hand side, lambda.
Yeah, it should be doable.
Coming up against a wall.
A wall. Wall of death.
Yeah, it's within 20 meters.
That could be dangerous.
And I'm already at almost 20 meters delta.
Roger. It's only 7 meters left on the ship, so. Can you open the
Yeah.
box for me?
Forward.
Forward box, yeah.
Copy.
You can stop her up there. That's, or whenever she's finished this move, that'll be it.
Data, is that sample 019?
That is correct.
Thank you.
Left side.
Roger, the left side.
Perfect.
Nice
Okay,
one.
close that box for me.
We have another question for the ROV pilots. Whenever you're ready.
Yep.
What are the visual clues that give you hints that rocks will be easy or difficult to move?
I guess I'd start that, Brandon. What makes you pick a rock that you want them to retrieve for your move?
I also look for them just to be loose. You can kind of see the sediment builds up around them.
Okay.
And there's a lot of these pillows are at least somewhat connected. And if you see some kind of disconnection, maybe there's a chance. And if they have the angles like we were talking about, those angular vertices, then it's a good chance it's a basalt, because that's how basalt naturally forms angular.
Can you press 5 on the bubble cam there for me? Cool, thanks. Okay, you can go back to 1.
I short-stopped your question, Dan. Do you have any secret tips of how you guess if a rock is loose or not?
None whatsoever.
What'd you see?
Just poke it.
I just
See
poke
what
it.
Poke it.
I poke it
there
and
you
it
go.
moves.
There's your cup coral on the right-hand side.
Really?
Yep.
Where?
And first one I've seen of the dive, right here.
Oh, I have to go look at that.
That's quite the sponge that's laid over that rock at the top.
Oh,
Yellow.
wow. Is this the preview for the still cam? It looks
How
like
do
it
you
is.
see that stuff?
How to see the cup coral?
Yeah, there's no way I would have ever spotted that thing.
I have coral vision.
Totally. Porch light.
I wish I had that superpower.
You want to zoom in on that guy, Dan? That full zoom, is it? I'm light years away. Oh, there you go.
So we've got a black coral off to the left-hand side too, which looks like Umbellapathes. And this coral here,
Okay, can zoom
the
out.
Scleractinian stony coral solitary polyp. If I hazard a guess, I would probably say Juvenia. Most things that look like that are Juvenia, or at least something in the Flobellidae family. Juvenia is a very widely distributed genus. Oh, more big